Hello everyone, welcome back to my channel Bunny's Biology. Today we are going to explore one of the most elegant idea in molecular biology, the genetic code. So the genetic code is the set of codon rules that tells the cell how to read the sequence of nucleotides in mRNA. This is the nucleotide sequence in mRNA and convert it into the sequence of amino acid in a protein. So the genetic code refers to the instruction containing in a gene that tells a cell how to make a specific protein. So what is codon? Codon is a sequence of 3 nucleotides in mRNA. That means each codon is made of 3 nucleotide bases. So 3 nucleotides join together to form one codon, and each codon corresponds to one amino acid or gives a start or stop signal during protein synthesis. So we all know that a specific segment of DNA that corresponds to a gene is copied into a messenger RNA molecule by the process of transcription, and this mRNA serves as a temporary and portable version of the genetic instruction. Then the cell ribosome, this is the ribosome, read the mRNA sequence, this is the mRNA sequence, and convert it into a chain of amino acid. This is the chain of amino acid. So ultimately production a functional protein. But this protein consists of one or more macromolecular subunit called polypeptide, and polypeptide are composed of smaller building block called amino acid. So a polypeptide is formed when multiple amino acids are covalently linked by a polypeptide bond in a linear chain. So the every protein in our body is built from a smaller unit called amino acid, and each amino acid is encoded by a 3 nucleotide codon. But our body uses 20 different amino acid, and protein are made of 20 different amino acid. So cell uses 4 different nucleotide bases: adenine, guanine, cytosine and uracil in mRNA. So how can mRNA specify 20 different amino acids even though it contains only 4 nucleotides? So let's solve this biologically. First, if each amino acid were encoded by just one nucleotide, then what will happen? Then 4 possible combination can be made from one letter code. That is not enough. We need 20 amino acid. So then if each amino acid encoded by 2 nucleotides, then 16 possible combination can be made. Still not enough. Then if each amino acid encoded by 3 nucleotides, then 64 possible codon are made. That is more than enough to encode all 20 amino acid. So that's why the genetic code is triple bases. So this is the structure that shows 64 possible codon can be formed when each amino acid is encoded by 3 nucleotides. In the genetic code table, the first base of the codon, this is the first base of the codon, is read from the left side. For example, if your codon is AUG, so A on the left, and the second base from the top, this is the second base and this is the third base, this is the first base. So second base from the top means this is the U, this is the top second base, and the third base, that is G from the right, this is the G base, this is the third base. So when you combine all three, you land on the amino acid that codon encode. So AUG encode the amino acid methionine, and this is highlighted in green. AUG is not just any codon, it is the start codon, the official begin translation signal for the ribosome. So that's exactly how the genetic code is organized, and every amino acid is specified by a 3-letter code, that is UUU, that encode phenylalanine amino acid. So here first U, that is the first base of the codon, is read from the left side, that is U base, that is the first base. Next is the second base, that also U, that is from the top, that is U, and third base, that also U, that from the right, this is the right side, and this is the third letter. So that's exactly how the genetic code is organized. So now look at the red one, UUA, UAG and UGA. These are the stop codon. They don't code for any amino acid. Instead, they act like a period at the end of a sentence and telling the ribosome stop, the protein is complete. So this is the top signal and also called chain termination codon. So out of all 64 codons, 61 actually code for amino acid. So we call them sense codon. The remaining 3 codons are stop codons. They do not code for any amino acid. So here the coolest part, even though mRNA has only 4 nucleotides, adenine, guanine, cytosine and uracil, and combining them in group of 3 and give us 64 possible codon. So although there are 64 possible codon and cell uses only 20 amino acid. So the multiple codon can code for the same amino acid. So the genetic code is not just a table of codon. It has several unique properties that make it one of the most elegant and reliable information system in biology. So the major characteristics are the code is triple code. That means each amino acid is encoded by codon and which is a sequence of 3 nucleotides. That means the code is triple code. So the genetic code consists of 64 codons and each made of 3 nucleotides. So out of the 64 codon, 61 specify one of these 20 amino acid used to build protein, and the remaining 3 codons act as termination signal. They do not code for any amino acid and instead instruct the ribosome to stop protein synthesis. So that's why the code is triple code. Next is that the code is degenerate and also called redundant. That means most amino acids are encoded by more than one codon. So we have 61 sense codon that specify the amino acid. We have 20 amino acid. So most amino acid have more than one codon. So that is the degeneracy of the genetic code. If you look over here, 6 different codon. This is the UUA, UUG, CUU, CUC, CUA and CUG. 6 different codon, all codes for amino acid leucine. So that's mean one amino acid codes different codon. Here is the leucine, one amino acid codes 6 different codon. So one amino acid have more than one codon. So that's the degeneration of the genetic code. And here is the some exceptional, that is AUG, which alone codes for methionine, and UGG, which alone code for tryptophan. And another example is the leucine that codes 6 codon, and serine also that codes 6 codon. Here is the 4 codon and here other is the 2 codon. Total 6 codon, and glycine also encode 4 codon. So one amino acid have more than one codon. Next is the unambiguous. The code is unambiguous. That means each codon specify only one amino acid. In previous we know that one amino acid have more than one codon, but here each codon specify only one amino acid. Like example, here UUU codon always code phenylalanine and UUC codon always code phenylalanine. So one codon never codes 2 different amino acid. That means UUU never codes 2 different amino acid. That means phenylalanine or leucine. Only UUU codon always code phenylalanine. So one codon specify only one amino acid. Even though one amino acid have multiple codon. Here GUU only code valine amino acid, never code methionine amino acid. So one codon only specify one amino acid, never encode 2 different amino acid. So that's the unambiguous. Next is that the code is nearly universal. That means same codon specify the same amino acid in almost every organism in earth. So a codon like UUU that encode phenylalanine amino acid, whether bacteria or a human. So same codon specify the same amino acid almost every organism on earth. Some exceptional in mitochondria in many organisms including mammals and other organisms use slightly modified codon, or protozoan tetrahymena also has small deviation in its nuclear genetic code. Next is that the code is non-overlapping. That means the code are read one after another. Like here example is the AUU, GCU and CAG. So these codon are read one after another, like AUU next GCU next CAG, not like AUU UUG, like UUG, not like UGC, UGC. So this is the overlapping code, and if you read this codon like one after another, like AUU, GCU and CAG, this is the non-overlapping code. So the code is non-overlapping. So this ensure a stable reading frame. This is the stable reading frame. During translation, the ribosome reads a code 3 bases at a time, and that moves exactly 3 bases forward to the next codon. So codon are read one after another, and the ribosome reads a code 3 bases at a time. Next is that the code is comma free or commaless. That means the mRNA is read continuously without gap or punctuation between codon. So there is no gap or punctuation between codon. Once translation begins, the ribosome moves codon by codon until it reaches a stop codon. The code is continuously without gap. Next is that the code has start and stop signal. So mRNA contains specific start and stop codon that signal the beginning and end of protein synthesis. So in both prokaryotes and eukaryotes, AUG, which code for methionine amino acid, is almost always the start codon for protein synthesis, and UAG, UAA and UGA do not specify an amino acid, and these three codon are the stop codon or chain terminating codon. So in mRNA, a stretch of nucleotide that begin with start codon AUG and end with a stop codon UAA, UAG and UGA without any interruption in between, and making it capable of being translated into a polypeptide that called open reading frame ORF, and this is actually protein coding region of gene that can be translated into a protein. So the genetic code may use only 4 letters, but with 64 codon it precisely specify all 20 amino acids. So it's universal, degenerate and unambiguous. So this simple code is the foundation of every protein our body makes. If this video helped you, make sure to like, share and subscribe for more lessons.